Today we're going to get into the subgroup analysis. So before I get into subgroup analysis, I just want to let you know that my personal philosophy of a single trial, a single clinical trial, individual clinical trial, I hope to discourage for too many subgroup analysis because sample size is not big enough. And usually subgroup analysis can be very misleading. All right, so my personal position is not to encourage too many subgroup analysis. So this book was published in 2020. Now, before I get into my points about discouraging or encouraging subgroup analysis, I want to give you some examples as to a successful experience of subgroup analysis. So this happened in the earlier part of my career. We were working on a drug for treating rheumatoid arthritis. So the drug we're developing was trying to treat rheumatoid arthritis and osteoarthritis. This happened in the 1980s and 1990s in those years. So in those years, the RA drugs are mainly classified in two major categories, NSAIDs and DMARD. NSAIDs being non-steroidal anti-inflammatory drugs. Aspirin, Naproxen, ibuprofen, diclofenac, many of these drugs are known as NSAIDs. They're non-steroidal anti-inflammatory drugs. And the other class is we call disease-modifying anti-rheumatic drugs. So there's these two classes. Now the difference is being NSAIDs are more for pain relief or symptom control, but DMARDs could hopefully slow down the disease progression. So NSAIDs are quick acting, symptom control, and more on pain relief. So if we think about rheumatoid arthritis, the two major symptoms is pain and swellness. So the joints are painful or joints are swelling or things like that. So most of the NSAIDs are very good in pain control, but they're not working very well in terms of slow down the swellness or inflammation. Right? The DMARDs are slow acting, usually 3 to 6 months. It helps slow down the disease progression. So NSAIDs are quick acting. Usually the first couple weeks, you're going to see the symptom improvement. The DMARDs takes a long time. So at that time, we're developing this test drug. We're hoping it to serve as an NSAID, so more on the pain relief, symptom control kind of things. And we designed a long-term protocol to compare the test drug against Naproxen. Naproxen is a known NSAID in treating patients with rheumatoid arthritis. It's a 5-year study with primary time point at 6 months. So our primary analysis is at 6 months. And randomized with stratification on DMARD usage. So in other words, for RA patients who entered the study, they either were on DMARDs or they were not on DMARD. All right. So if they're on DMARD, they're supposed to be on stable DMARD. And during the whole 5 years, they're not supposed to change it. If they're not on DMARD, then they're not supposed to add DMARD during the whole 5 years. So these are the DMARD naive patients or DMARD experienced patients. So we stratified on this basis. So it's very justified to sort of perform subgroup analysis, look at patients' response, comparing test drug against Naproxen from the DMARD users versus DMARD non-users. Primary endpoint at that time, that was in late 80s, I remember. The primary endpoint was a physician assessment of disease activity. This is a 7-point scale, a 5-point scale. It's no symptom, mild, moderate, severe, very severe. Of course, we coded as 1, 2, 3, 4, 5. If you look at the mean change, the test drug improved about 0.76 unit. Naproxen improved about 0.57 unit. With p-value of 0.0103. All right. just imagine, all right, the improvement from very severe to severe, from severe to moderate, or from moderate to mild, or something like that. That one level improvement, if you look at hundreds of patients, look at the average, a 0.76 is fairly meaningful improvement. Naproxen is a known NSAID. We already know naproxen can help with patients with RA, and their improvement is 0.57 units. That's already sort of good enough for that drug to be approved and marketed for many years. Now, so the test drug is better than naproxen with a small p-value. And because we stratify by DMARD usage, so it's natural to look at subgroup analysis to look at DMARD users and DMARD non-users. And let's take a look at this bar chart, all right? So you look at the DMARD users, the test drug is better than naproxen. The DMARD non-users, the test drug is better than naproxen. That's pretty clear, all right? But if you look at closer, you can see for the DMARD users, improvement is about probably 0.1 some unit, all right? Now, the non-users, the improvement is about 0.2 some units. So this difference, the treatment difference for the non-users, apparently is greater than the difference on the users. So we look at these results and we say, hey, the test drug may have a DMARD characteristic. So for the DMARD non-users, they can improve much better. So the test drug could potentially be a DMARD, all right? Test drug has better efficacy than naproxen. Results from subgroup analysis indicate the test drug could be a DMARD. Further development demonstrate DMARD efficacy of the test drug. So change the direction of development based on subgroup findings. So it's just a subgroup finding. You can change the direction of further development. So subgroup analysis sometimes can be very useful, can give you a very good hint as to which direction the development program should go. All right. So that's my first example to show where subgroup analysis could be helpful, could be useful. So as you recall, we talked about intention to treat analysis. Analyze as randomized, including all subjects. Well, it's important to include all subjects. Imagine a wonder drug cures 9,999 out of 10,000. One died. We cannot exclude this death, right? We cannot treat it as an outlier. We should report all data and analyze as randomized. So that's the intention to treat principle. All right. Now, the primary analysis for a study is typically intention to treat or full analysis set. Sometimes people using per protocol set, but per protocol set can be viewed as a subgroup already because you're excluding protocol violations. All right. But anyhow, these are the typical primary analysis set. Any analysis based on part of the entire analysis is considered a subgroup. For example, you look at analysis by gender, analysis by age, by center. These are all different kind of subgroup analysis. Now, why do we want to do subgroup analysis? All right. Now, to address particular concerns in some specific subgroups, right? I mean, at the design stage, you may have thought of these particular subgroups and you want to take a look at them. Or to explore whether the test drug is more efficacious or more harmful in the subset. To provide supportive evidence to the main findings, let me show you this first plot here. All right, this is one of the BI drugs we call Pradaxa. So Pradaxa, I think, was developed, I think the phase 3 trial was like 2007, 2008, 2009, in those years. It was comparing against warfarin. So Pradaxa is a newer generation of the anticoagulants. And we know warfarin is already marketed anticoagulants for, I think, several decades when we make these comparisons. So looking at, well, there were, I think, 3 treatment groups, 2 doses, Pradaxa 150 and Pradaxa 110 versus warfarin. We're trying to show non-inferiority, all right? So you want to be right around 1 to be non-inferior, all right? So on this side, warfarin is better. On this side, Pradaxa is better, all right? So the entire study is 18,040 patients. I think Pradaxa 150 is about 60,000, and warfarin is about 60,000, and Pradaxa 110 is about 6,000. All right. And so if you look at all patients, so here we're looking at the major bleeding events. All right. Major bleeding. You look at the comparison of Pradaxa against warfarin. With about 6,000 patients, and Pradaxa there's 350 major bleeding, and warfarin there's 374 major bleeding. So the point estimate is a little less than 1. So you look at I think it's HR hazard ratio. If you look at the hazard ratio, that 350 is a little lower than 375 in terms of hazards, and look at the ratio is less than 1, below 1. All right. So in other words, if it's less than 1, it's in favor of Pradaxa. If it's greater than 1, it's in favor of warfarin. All right. So that's the overall patient. Now you look at the vitamin K usage. Vitamin K usage, there are about 3,000 users and 3,000 non-users. So these are non-users, naive and experienced. Naive and experienced. And then you look at the hazard ratio, all right? So 1 is a little, all right, the naive patients is about 1.0, the experienced patient is about 0.94, with these are the confidence intervals, so, as you can see. All right, so you look at all the subgroups. You look at age, you look at gender, look at weight, look at history of stroke, history of TIA, and let's see, CHAS score, creatinine clearance, region, aspirin usage, and you look at all these subgroups, and they give you a consistent pattern. Of course, you can say this is, but if you look at sample size, it's only 14 events out of 1028. So this, and you can see a very wide confidence interval. All right, but by and large, most of the subgroups support the main finding. All right, so to provide supportive evidence to main findings. All right, so the reasons are address particular concerns in the specific subgroups to explore whether the tetralogue is more efficacious or harmful, support main findings, and to generate new hypothesis of drug effect. The first example I showed you about this DMARD usage that give you a new hypothesis, right? And sometimes to address regulatory queries. So FDA or EMA may have some interest about particular subgroups, so they will perform subgroup analysis. So there's some guidance, and this New England Journal of Medicine 2007 says investigators frequently use analysis of subgroups of study participants to extract as much information as possible. Such analysis may provide useful information for the care of patients and for future research. However, subgroup analysis also introduce analytic challenges and can lead to overstated or misleading results. All right. If you look at ICH E9, it says when exploratory, these analyses should be interpreted cautiously. Any conclusion of treatment efficacy or lack thereof, or safety based solely on exploratory subgroup analysis are unlikely to be accepted. So, in other words, subgroup analysis is not highly encouraged. I mean, at least from my perspective, you're dealing with a single trial. To do too many subgroup analysis may not necessarily give you the information you want. All right. So the basic thinking is that whether are we doing a confirmatory practice or exploratory practice. Hypothesis testing is confirmatory practice. All right. Now, subgroup analysis of basically hypothesis generating. You're not testing a particular hypothesis, you're generating a hypothesis, because by looking at subgroups, you may have a different conclusion or something that will lead you to do something different. All right, but that is not a decision. All right, hypothesis generating, you're just raising questions. It's not giving you a clear direction which way you should go. Inferential statistics versus descriptive statistics. My personal inclination is for all subgroup analysis, I provide descriptive analysis. I will not provide inferential statistical analysis. I will definitely not will give p-values, because p-values can be very misleading. All right. Now, for the subgroup analysis, are a regular discussion or change in label language, or just of scientific interest. So before performing any subgroup analysis, I should ask all these questions and try to have a good understanding of what we want to achieve before I perform any of the subgroup analysis. And of course, design feature versus post hoc. I mean, people usually take the design feature more seriously. I mean, the first example, we talk about DMARD usage because we stratified on it. So there is something that of interest at the design stage. So then the subgroup analysis seems to be justified. Learning by nature is an exploratory practice, all right? So most subgroup analysis give us a learning opportunity, but it's not a decision making. Right. So you can perform many subgroup analysis. Is that a learning, or is that data dredging? Or sometimes I would say, or we torture the data until it confess. Okay, so doing too many subgroup analysis from a single trial, it probably doesn't give you too much information about the properties of the drug. Be clear that this is not confirmatory. All right, subgroup analysis by nature are not confirmatory. I mean, of course, the enrichment design is a different story. All right, but for most of the trials, if you didn't pre-specify particular subgroups, those subgroup analysis, the findings and stuff like that could very well be misleading. If it turned out that only one of the subgroups demonstrated efficacy, the regulatory agency may restrict label. So what I mean is that if you look at the subgroup by gender and look at the test drug to placebo difference for male versus for female, if there is a strong efficacy on the male side, that could be because the efficacy in the female side is not strong enough. And if FDA sees this, they may say, all right, this drug may be only good for male. All right, so there could be a potential market issue kind of things. Post-hoc analysis is in most of the time they do not carry much weight. All right. Overall treatment effect is significant in the complete study. So confirm consistency across major subgroups. That's what that first plot did, right? Give you some level of consistency. Identify safety problems that are limited to a subgroup of patients. So we may want to identify certain subgroups where they're more likely to have certain kind of adverse events. So there are many issues about post-hoc analysis. All right, post-hoc versus pre-specified, often unclear how many were undertaken, whether some were motivated by inspection of the data. And a subgroup is respectable and worthwhile when established a priori from pathophysiological principles. All right, so the pre-specified analysis carries some weight. But post-hoc analysis usually doesn't carry much weight because it could be just purely data-driven. You look at the data, you're curious, you do something else, but that does not give you a definitive approval or not approval, or whether the drug is really good for the subgroup or not. We don't have that. All right. When I was at Pfizer, we worked on amlodipine. Amlodipine is, I think, the it was developed for controlling blood pressure. It's a calcium channel blocker. All right. So in 19 was the study was a few years before 1996. I think it was 1993, 1994, something like that. We did this PRAISE trial. PRAISE. I think amlodipine was approved in 1990, so this was post-marketing study. And we're trying to see if amlodipine can also help patients with stroke, okay? So the pre-study was amlodipine versus placebo in 1153 patients. Predefined stratification factor was ischemic stroke or non-ischemic stroke. And you look at the PRAISE study, you look at the mortality just on the non-ischemic patients, p less than 0.01, 0.001, all right? So this is PRAISE study, all right? At that time, we only designed PRAISE, but later we designed PRAISE 2, so we'll come back and call it PRAISE 1, all right? So look at the mortality for the non-ischemic patients that amlodipine is superior to placebo with a p of less than 0.001. So it means something that seems like the non-ischemic patients could benefit more from amlodipine. So the textbook approach is that if you see some signal, you try to repeat it. Can you confirm that for the non-ischemic patients, they can benefit from amlodipine? So study was repeated in PRAISE 2, so we call it PRAISE 2, and then we came back and called the first study PRAISE 1. So repeating PRAISE 2, enrolling non-ischemic patients only. So we are only looking at non-ischemic patients. Then here's the results. It's not significant. And actually the p-value is slightly greater than 1. So that's a relative risk. So you're looking at the PRAISE study where indicates that amlodipine could help the patients. And then you repeat it, you couldn't find it. So in most cases are like this. You look at something from the subgroup, you thought you have some signal, and then by repeating it, the signal is gone. So there were some publications, all right? Post-hoc analysis, there's lack of power. I mean, there's basically no power because, of course, we have to come back with the concept of power. Power means if the drug works for any subgroup, and we did not have that sample size calculation to do that. All right, lack of power and chance of missing significance effect in the subgroup, just a function of size. I mean, your sample size is not big enough. All right. Antihypertensive treatment for primary prevention is ineffective in women, so that's another publication. There's some other publications. So, in other words, the post-hoc analysis don't really have the kind of status as a pre-specified analysis. All right, so before looking at subgroup analysis, it's natural to look at the treatment by subgroup interaction. All right. Do treatment effect differ at each level of factor? So what do I mean? All right. So if I stratify by gender, male and female, and I'm comparing test drug against placebo, now I have 4 means, right? Test drug mean for men, test drug mean for women, placebo mean for men, placebo mean for women. All right. So I got 4 group means. Now, if it turned out there's no treatment effect, there's no gender effect, then the 4 means should be about the same value. All right. So there's no treatment effect, no gender effect. What if there's treatment effect but no gender effect? All right. There's treatment effect. So test drug is better than placebo. Test drug is better than placebo. And there's no gender effect. So for male and female on placebo, their mean is about the same. For test drug, male and female, their mean about the same. So this is the case when there's treatment effect, but there's no gender effect. And this is the case when there's gender effect, but no treatment effect, right? Test drug and placebo are almost on top of each other, right? But male response is higher than female response. So this is a gender effect but no treatment effect. What if you have both? Here's the case you have both. You have both treatment effect and gender effect. Right, but this is a case without interaction. All right, so you can have a treatment effect, you can have a gender effect, but that doesn't mean you're going to have an interaction. And statistically speaking, we call this parallelism. All right, so these two line segments are parallel. Now, if there's interaction, there could be this kind of interaction. We call it quantitative interaction. So in other words, test drug is better than placebo in both male and female, but the amount of improvement for one group is different for the other group. All right, so these are two subgroups. All right, male subgroup and female subgroup. And you look at treatment effect of test drug against placebo. All right, and they're not parallel, so they have interaction. All right, but this interaction is what we call quantitative interaction. We want to differentiate from this situation. This is quantitative interaction. In other words, the test drug is better in male, but it could be harmful in female. So if the treatment means goes a different direction, that is qualitative interaction. And here is quantitative interaction. So that's what I mean by interaction. All right. The treatment effect differ at each level of the factor. All right. Is a treatment placebo comparison different for male or female patients, for older or young patients, for any other subgroup factors? All right. For example, is a test drug better in male and worse in female? So that's the qualitative interaction. Right. If the test drug is better than placebo in both male and female, there should be no interaction. And of course, we're going to look at regions, geographic areas, other stratification factors. We can do this because it's justified, because it's pre-stratified. Okay. Nowadays, we don't talk too much about multi-center trials because most studies today are multi-regional trials. But back in the 80s and in the 90s, we do a lot of multi-center trials. All right. So you look at center as a stratification factor, and by center analysis commonly applied, and treatment by center interaction typically not significant for an active treatment. We just use descriptive statistics. And this is what I used to do, but nowadays center become less important in your model. Okay, so that's a brief introduction about subgroup analysis. All right. But nowadays, we not only do subgroup analysis, we also do something we call subgroup identification. So from the study results, you try to identify potential subgroups so that for future trials, you can take advantage of this understanding. Or you use this to select patients to see which patient will benefit most from your drug, or which patient could be is more likely to develop adverse events or safety concerns. All right. There are basically three types of markers. There's surrogate markers, there's prognostic marker, there's predictive marker. Okay. Now, surrogate marker is correlated with primary endpoint. All right. So you can look at the surrogate marker change. Now, in a way, if I want to do a study to lower blood pressure, of course, blood pressure itself is my primary endpoint. All right. But if I want to design a study for cardiovascular disease or cardiovascular conditions, then blood pressure becomes a surrogate. Right. So the blood pressure may be correlated with the primary endpoint, with cardiovascular events. All right. And we can see changes of the surrogate marker over time, what patients on treatment, kind of things. All right, that's surrogate marker. Now, baseline characteristics are mostly used for prognostic marker or predictive marker. So we look at the baseline characteristics of patients, and we try to see will the patient likely be more responsive or less responsive to the test drug. All right. So if we're talking about more responsive, less less responsive by itself, we call prognostic markers. But if it's specific to your test drugs, that's predictive markers. All right. So predictive markers are related to the test drug itself. All right. Markers can be non-prognostic, non-predictive, can be either or both. All right. Predictive markers were used to develop target therapy. So if we have some predictive marker, we may want to take advantage of this predictive marker to help us design target therapies. For patients with a certain characteristics, we want to have the drug so that these kind of patients will benefit most from the drug. All right. So you select the marker positive patients, and subgroup identification is about finding markers. So when we talk about subgroup identification, the process about finding which marker will give you a good differentiation of your treatment benefit. So design considerations. Of course, there's enrichment designs. All right, for enrichment design, what you do is that you look at marker positive patients. All right, you probably recruit all comers, everybody with this target disease, and then you select only the marker positive patients and look at them as a stratification factor. So that's one strata, and then with marker negative as another strata. All right. So enrichment design would be you recruit all comers, and then your primary analysis, your primary interest is only a subgroup of the all-comers, not the entire all-comers. And that subgroup was defined based on this marker positive measurement. All right. So that's enrichment design. Primary objective can be the entire population. All right. Or it can be the marker positive patients. So that's the other way around. You can either show that the drug is good for all comers first, and after that, you try to demonstrate this test drug is superior in the marker positive patients. That's one way of doing it. The other way of doing it is you want to first show the drug is very effective for marker positive patients. And after you were able to demonstrate that, then you say this drug could also be good for all comers. All right. So study objectives must be clear. All right. And nowadays, popular trials are MRCT, multi-regional clinical trials. All right, multi-regional clinical trials. You're doing a global trial, and you divide the global countries into certain regions, and then you have this multi-regional clinical trials. Well, MRCT is more popular after the turning of the century because globalization and modernization. And it's getting to be more popular in 21st century. And so if you're doing multi-regional trials, region is a natural sample factor. You can use either region or country, all right? And that'll affect your study conduct also. All right. Now, regular approval, of course, different countries may have different settings. All right. So there are a lot of design considerations in designing multi-regional clinical trials. All right. MRCT. So before you design the MRCT, you need to have a clear definition of what does it mean by region. All right. Is that a country, or is that many countries together? Or are these countries are they geographically consecutive, or they're geographically isolated? All right. So what is your definition of region? All right. It depends on the trial. All right. Could be country, could be a combination of several countries. This definition impacts study design and data analysis. All right. So this definition of multi-regional design, what does it mean by region? And that definition impact on study design and data analysis. All right. For example, if region is considered as a random effect, then a large number of regions is needed, and it's less powerful. Okay. So if region is considered as a random effect, a region can be considered as a fixed effect, right? If you consider it as a fixed effect, you can say region 1 has this kind of mean, region 2 has this kind of mean. That's a fixed effect. But if they're random effect, then you need large large number of regions. So in MRCT, there could be an analysis model how you analyze the MRCT data. And in MRCT, we're looking at consistency assessment of regional treatment effects. So if test drug is better than placebo in one region, it should be about the same in another region. All right. And of course, in designing MRCT, you need to consider the regional sample size allocation. All right. And you're concerned about the results interpretation. All right, region is a surrogate marker for many intrinsic and extrinsic factors. All right. So although we're looking at region as potential subgroup, but each region itself could be different from the other region in terms of smoking status or lifestyle kind of things. So region can be a surrogate for many intrinsic and extrinsic factors. Region could be representative, all right? Such factors as race or ethnicity, disease epidemiology, background therapies, and medical practices, and these could be considered as potential factors for MRCT. And there's regional regulatory requirements, all right? They could lead to different margins for non-inferiority trials. So there are region-to-region concerns. All right, for MRCT, how do we calculate sample size? There are certain agencies that they require a certain amount of patients to be in their country or to be in their jurisdiction in order for you to complete the MRCT. All right, sample size calculation. And you look at overall treatment effect, you look at regional treatment effect, and then you look at between region vulnerabilities. And these models, these MRCT models, they can consider region as fixed effect, as a random effect, and as a discrete random effect. So you can model it differently depending on which model you're assuming the MRCT follows. And we need to know the local regulatory requirements, and we want to know practical considerations. All right. So for MRCT, there's overall sample size, and there's sample size allocated in each region. All right. So that's sample size issues. And for analysis considerations, overall, and subgroups by region, by country, by regulatory agency. So you can have overall efficacy or overall safety, and then you look at the subgroup efficacy and subgroup safety. Ordinary subgroups like gender, disease, severity, and things like this. All right. So, so that's a multi-regional clinical trial concern. All right. Now we'll get into a little bit about subgroup identification. All right. Could be based on failed phase 3 trials, or could be based on successful phase 3 trials. All right. So after phase 3, whether the drug is successful or not, you can look at subgroup analysis because phase 3 gives you a very large database. All right. So if you're dealing with this situation, you want to know which candidate markers you're going to select. All right. We're going to have some understanding of how we select the markers. And selection of subgroups search parameters. So when you do a subgroup identification, you need to define a subgroup search parameter and covariate adjustment in analytical models. Right. These are some of the practices for subgroup identification. Oops. Most subgroup identification approaches are based on decision trees. So the next few slides will just briefly introduce some of the subgroup identification techniques. And most of them based on decision trees. They're either regression tree, the response is continuous, or they're classification tree, where the response is categorical. All right. Now, the subgroup identification is a binary splitting the data set. So the overall data, and then chopped into two halves, all right? And they grow a large tree. These two halves could be young patients or old patients, or male patients versus female patients, or background drug users, background drug non-users. So you can do a binary splitting of the data set. And then after you keep splitting, you're going to have a tree structure, all right? And after you have this tree structure, then you do some pruning, all right? Pruning is certain criteria, try to reduce to smaller trees. So you start out with binary splitting the data set with many factors, and you grow a large tree, and then you sort of prune and shrink to a smaller tree, all right? Or we can call it subtrees, all right? To grow many trees and use a random forest to average across the many trees, okay? So in subgroup identification, we sort of do a binary split, and we're trying to grow a big tree, and then we prune it, and then we grow another tree, and we prune it, and then we'll grow another tree, we prune it, and then we can grow many trees. So after we grow many trees, and then we can use the random forest to average across many trees. So we always start with interaction trees. What is interaction tree? So in other words, as you recall, there's interaction, the interaction term that the test drug is better than placebo, and then you look at you look at one group, the test drug is better than placebo with a significant amount, and the other group, the benefit is not that clear. Now, so these are, so we call it interaction trees, all right? You're looking at interaction between treatment and covariate. In other words, when you split, you're not looking at treatment effect of the overall population, you're looking at treatment effect within each subgroup, right? So if you go back to here, interaction is the difference between these two treatment differences. Right? There's a treatment difference for male, there's a treatment difference for female. And what are those differences? The difference of these two, that's interaction, okay? All right. Let's see. These are interaction trees, all right? So interaction between treatment and covariate. In particular, the data into two subsets was the greatest interaction. So you look at either gender or age or background medication usage, so you identify these, all right? And grow a large tree and then prune, all right? So you start with a interaction tree, and you just grow to a very large tree, and then you prune. Virtual twins is another concept. So this idea is this, all right? If you run a model, all right, we have only two treatment groups, test drug versus placebo, all right? So you start with interaction tree. So you build up this tree, and then you come up with a statistical model to perform the analysis, all right? So you start with interaction tree and select a model based on Akaike information criteria, and using this model to predict response of ICE subject. All right. So here's what we're doing. All right. You build a tree and you come up with a statistical model. This model will have all the covariates necessary in the model. All right. And now you have the ICE subject. The ICE subject, the first, second, whatever. So you have this subject. Now, this subject may actually be on test drug group or may actually be on placebo group. It doesn't matter, all right? So what you do, you take the subject, given the subject's age, gender, race, and the background medication, all the characteristics used in your model, okay? So now, you try to predict the subject's response by plugging a test drug. So you already have the patient's race, gender, and all subgroups, all these factors in the model. And now, if the subject is treated with test drug, you plug in test drug, you're going to have a point estimate. Even though the subject was treated with test drug, you can also assume if the patient receives placebo, right, given the same kind of covariate structure you have. Now, suppose there's a placebo effect for the subject. Now you have this two effects. One effect being the subject's test drug, another being the subject's response while the subject's on placebo. So now you have these two differences. And these differences would give you the virtual twin kind of analysis. So that YIT and YIC represents the response of ICE patient treated with test drug or control. And define the difference. So for the same subject, you try to predict the subject's response while under treatment, under test drug, or while under control. All right, and then you have these two observations. You take the difference. The difference is denoted as DI, D sub I, which can be regarded as a treatment effect of subject I. So that's the treatment benefit of subject I. If the subject I is treated with placebo, here's the response. If the subject I is treated with test drug, here's the response. And you define, all right, you define something, you define D such that all the ZI is greater than D. And the formula is set A. All right. So now you have this virtual twins analysis. Of course, you can do continuous response variable or discrete response variable. Okay. And size is another way of subgroup identification. Subgroup identification based on differential effects, differential effects search. You call it size. All right. And there's three components. So you break down your data set into a training data set. All right. And you identify these candidate subgroups. And then you go through the subgroup identification. After that, you take that algorithm and apply to the confirmation data set, the use of test data set. So there's training data set, there's test data set. Okay. And size can also adjust for alpha. All right. Guide is another subgroup identification method. Generalized unbiased interaction detection and estimation. Okay. You consider both interactions and residuals. So in this analysis, you look at interaction. I mean, most trees are interaction trees. All right. So here they're not only considering interactions, they're also considering residuals. Y is the response, Z is the treatment, X is the covariate, and you can look at Y being binary, both X, Y, and continuous X is binary. So you can do that. So that's pretty much an introduction of subgroup identification. The trial is powered to study entire study group, so there's not enough power to look at subgroup. There is insufficient power to make sure treatment comparison in any subgroup. Again, in my subgroup analysis, I never give p-values. Right? Computing p-values within a subgroup is not appropriate. If there's a signal from subgroup analysis, design a new study to test this hypothesis. In certain situations, the primary interest for a specific subgroup at the design stage, that's in enrichment design. So you're only interested in the marker positive subjects, okay? The study should be powered for that subgroup, and stratification is recommended. Pre-specify the primary analysis based on subgroup. All right, so the next concept is very important. Subgroup need to be defined at baseline. So post-baseline characteristics cannot be used as subgroup characteristics. Right. Post-baseline characteristics may be affected by treatment. So you cannot use post-baseline data to classify subgroups. All right. Now, if the subgroup is defined at baseline, so there's a treatment by factor interaction, all right? And then you have the response. You either have treatment response, you have factor response, you have treatment by factor interaction response. But for post-baseline, you cannot say that because the factor was affected by treatment already, and then you see the response, all right? So in this case, treatment effect is not interpretable. Let me give a compliance data example, all right? So we were in a trial team meeting. So the results are not looking good. So patient response is not as good as we expected. And of course, we have very poor compliance. So the clinical colleague stood up and says, "Nighty, you look at these patient responses and the poor compliance." And then she said, patient did not respond because they did not comply. And I said, patient did not comply because they did not respond. So there's no causal relationship, right? So patient compliance data, you cannot use that as an analysis tool for your response, for your efficacy. That can be misleading, right? So for intention to treat, analyze as randomized, all right? So I want to give you a Naproxen example, all right? Now, Naproxen was developed to treat AMD, age-related macular degeneration. The primary endpoint is change in visual acuity at 54 weeks post-baseline. And the primary efficacy endpoint is to see if the patient loses 15 letters or not. Okay, I think I probably have this eye chart, all right? So this is ETDRS. It's ETDRS, I think is early treatment for diabetic response scale, something like that. All right, so you have this eye chart. The patient will look at the number of letters, and then you have the baseline number of letters, and after 54 weeks, you look at the number of letters they can see. Now, if they lose 15 letters or more, that's considered a non-responder. If they lose less than 15 letters, they're considered as a responder, right? So your response, your analysis is based on proportion of responders of test drug versus placebo. Now, at that time, the only available, so Naproxen was the first pharmaceutical treatment for AMD. So before Naproxen, all they have were PDT. PDT is photodynamic therapy. Photodynamic therapy is basically a kind of dye. You sort of inject the dye into the patient, and the dye goes through the circulation. Then the dye, you can see it from the back of your retina. And they use X-ray to focus on these dye area to kill all those photoreceptors. So that's PDT, okay? Now, we're running Naproxen trials, phase 3 trials. There are 3 types of PDT usage. There's prior PDT, so some time ago, this patient's study eye received PDT treatment, all right? Or baseline PDT, so this patient during the baseline stage, the patient received PDT, or post-baseline PDT usage. So we completed the study, we submitted the NDA, the new drug application, and then we're trying to prepare for advisory committee meeting. And the clinical team says, "Nighty, I want to see subgroup analysis." So I tried to convince them, yeah, to look at the prior PDT or baseline PDT, that's reasonable. But post-baseline PDT, you cannot use it as a stratification factor. All right, but they still want to see it, so I have to come up with a way to convince them why not to use it. All right, so you look at the data for prior PDT usage and baseline PDT usage. There's no treatment by PDT interaction. There's no interaction. But for post-baseline PDT usage, Naproxen treated patients did not receive more PDT than sham patients or placebo patients. So in other words, the Naproxen efficacy cannot be explained by overuse of PDT because Naproxen patients did not receive more PDT. Okay. So they want me to do a post-baseline PDT subgroup. I say you cannot do that because estimate of treatment response in the presence or absence of post-baseline PDT is biased. So here's what I did. I did a graph. All right. Remember, the primary efficacy endpoint is responder yes, no. All right. So I'm only looking at the placebo patients. All right, I'm only looking at the placebo patients. And I look at the placebo patients who received post-baseline PDT and who did not receive post-baseline PDT, right? So these are only placebo patients. Now, if you look at the responders, the proportion of responders, 25 out of 62 is just a little bit less than 50%, right? Maybe about 40% or so. But 139 out of 234, we got close to be 60% response rate. And you see, patients used post-baseline PDT used post-baseline PDT, their response is worse than patients did not use post-baseline PDT. Can you say that post-baseline PDT is harmful to patients? You cannot say that, because the placebo patients, their vision deteriorates so much that they require PDT. So they're already deteriorated too much, then they added PDT. If they did not deteriorate that much, they don't need PDT. So you cannot say there's a causal relationship to say PDT causes the patient response worse. You cannot do that. All right. So I use this chart to convince the team that post-baseline PDT is not reasonable. It's not justified. So how do you do subgroup analysis in combined studies? So you combine many groups, many trials, so ISS being integrated summary of safety, integrated summary of efficacy. You got a large enough sample size, you may be able to do some subgroup analysis. After pooling across studies, there may be sufficient sample size. All right, combined analysis. A clinical trial is designed to study the entire patient population. Subgroup analysis are mainly exploratory for exploratory purposes. If there's a need, it is desirable to pre-specify at the design stage. Results obtained from subgroup may not necessarily be replicated. Potential consequences of restrict labeling. Okay, thank you.